Next, we're going to spend just a little bit of time talking about the contraction of muscles. So a muscle cell is the same thing as a muscle fiber. So whether you, just to kind of help clear up any terminology. When skeletal muscle cells are viewed under a microscope, they'll have distinct bands called striations. These are going to be formed by an arrangement of myofibrils within the cell. Myofibrils are specialized bundles of protein, and each one contains two types of myofilaments. One is going to be myosin, or thick filaments. The other will be actin, or thin filaments. Those are going to be much more numerous when they're looked at under the microscope. Here's an example of what that looks like. So here we have a section of a muscle fiber. Here is that striation that you can see. So skeletal muscle is going to be very easy to identify under the microscope due to those heavy striations. But these different lines or these different striations are indicated by the actin and the myosin filaments that are going to be found in the muscle. The myosin head, it's also known as a cross bridge. It's going to be attached to the nearby actin filament. What's going to happen is the head is going to bend and swivel. It'll pull the actin filament towards the midline of the sarcomere, which will be the center line down that muscle fiber. So here is going to be our sarcomere right down the center. What's going to happen is that these myosin heads are going to latch on to this actin filament and pull it towards the middle, and that will happen on both ends. And so what we're going to see is a contraction from both sides coming in towards the center. So as we're pulling towards the midline of the sarcomere, the myosin head is going to disengage from the actin filament and then grab again and pull, and grab and pull, and grab and pull. And it's going to work sort of like a ratchet system, where it's going to pull those fibers a little bit at a time until it's fully contracted and then release. The movements of myosin is going to require ATP. Every time we grab and pull and grab and pull and grab and pull, another molecule of ATP is going to be required. Once we've grabbed and pulled all the way towards the midline of the sarcomere, we're going to relax. The muscle actin filaments will slide apart, and then we begin again. Muscle contraction is going to be controlled by the availability of calcium ions. Muscle cells contain the proteins troponin and tropomyosin, and the troponin-tropomyosin complex and calcium ions ultimately regulate muscle contraction at the actin-myosin binding sites. So they are going to be what's responsible for pulling those heads across. Here's an example of what that looks like up close. We have our troponin and our tropomyosin complex. That's going to be what is found on the outside of the actin filament. This is where we are going to latch on and attach that myosin head. Calcium ions permit the contraction by causing the binding sites on the actin to be exposed. Once we expose those binding sites, those myosin heads can reach up, grab on, and pull that, form that cross bridge and pull the muscle fibers across, or pull the muscle fibers into contraction. An example of a homeostatic balance that may be found within the muscular system might be rigor mortis. Rigor mortis is caused by muscle contraction as long as ATP is present. Without ATP present, the cross bridges can't be broken, and so the muscles stay contracted. Within 3 or 4 hours after death, the muscles are going to become very stiff. Rigor mortis will set in. Eventually, given enough time, actin and myosin will gradually break down. The muscles will start to relax again after 2 or 3 days. So rigor mortis is a temporary condition that happens as long as those cross bridges can't be broken. As long as there's not an immediate production of ATP and before the muscle fibers themselves start to break down and deteriorate, we're going to find rigid muscle contraction. Muscular dystrophy is a group of inherited conditions in which the muscles weaken. If too many calcium ions enter a muscle cell, the proteins may be destroyed, eventually causing the cell to die. On a large scale, the muscles are going to weaken. Another homeostatic imbalance of the system would be Duchenne muscular dystrophy. It's one of the most common forms of muscular dystrophy. What happens is there's a gene that lacks the production of the dystrophin. The gene is defective, and thus it doesn't produce the correct folded protein dystrophin. The lack of that dystrophin will allow excess calcium ions to enter the muscle cells, and it will eventually poison and kill those cells off.